Hello, and what we're going to do this week is focus on immune system responses, both the innate and adaptive immune responses, and some of the applications of immune responses. This is kind of a segue to get you prepared for what we're going to have are the host pathogen interactions, and then get you into epidemiology, and eventually we're going to be getting into, yes, I know you can hardly wait, all the different types of diseases. But first, what you really have to do is understand some of the aspects of how the body fights against various types of pathogens, and this will also give you insights into some of the mechanisms of how vaccines work and how some pathogens are able to evade that immune responses. As always, I would encourage you to review the terms, the key terms on page 361. We're going to be going through basically chapters 14, 15, and 18 here. So you really do need your lecture notes with you. I also encourage you to review the Glimpse of History, which is really one of the earliest cases. Melkinoff, with his work done very, very early, became sort of the early pioneering efforts in immunology. Now, when we talk about innate immunity, this is the routine protections against pathogens existing from birth. It's passed down from one generation to another. It consists of three primary components. And you can see here there's white blood cells, red blood cells there. But what we have to talk about are the first line defenses, which prevent or block microbial entry. This includes such structures in our body as the skin, mucosous membranes, and also the antimicrobial substances present. Then we have the sensor systems, which detect damage and microbial invasion. These include pattern recognition receptors, PRRs, and the complement system. And finally, we have the innate effector system, which is to eliminate the invader. This includes interferon response, phagocytosis, complement activation, inflammatory response, and fever. That's what a lot of what we're going to be facing in this chapter. When we go into chapter 15, we're going to be dealing with the adaptive immunity, which develops throughout our life, and it's the result of exposure to microbes and other foreign material. So this enhances the ability of the host to defend itself against microbial attack. To give you one type of perspective, the innate always exists with us. The adaptive gets altered or tuned as we grow and develop, depending on how we're exposed to different antigens, different pathogens, etc. The antigen is a substance that causes the immune response. Antibodies are part of the adaptive immune response. They produce these Y-shaped proteins that bind to antigens and target them for destruction or removal by other host defenses. Now, you need to keep in mind this, that the adaptive immune responses not only can destroy host cells infected with virus, but they will also be able to destroy host cells that may have other invaders, such as intraparasitic organisms. When we talk about the host cells, these are the self cells, the cells that belong to the host, basically your cells of your body. If you see the overview here, we have the first line defenses in this image, and these are the walls that prevent entry of microbes into the body. These are the physical barriers, and they block entry into the skin, mucosous membranes, as well as the antimicrobial substances that destroy organisms prior to entry. Here's one thought. Why are third-degree burns, when we talk about this, the extent being 30 to 50%, why are third-degree burns so fatal to the body? Basically, the loss of the dermal layer makes entry of pathogens very, very easy. This is basically the loss of the first link defenses, the first line defenses, I should say. Sensor systems. These function as monitors to invasion and tissue damage due to invading microbes. Select host cells act as sentinels, guardians, and they are located at specific sites, and they recognize microbes by pattern recognition receptors, these PRRs, and these are protein receptors, either on the cell membrane or on the inside of the endosome or phagosome or inside of the cytoplasm of the cell. They also allow for the phagocytosis and the destruction of the invader. Also, in blood and tissue fluid is the complement system. Now, complement system is a series of proteins. It acts in complement with adaptive immune defenses that upon activation lead to removal and destruction of invaders. Effector actions. These are tailored responses to different types of invaders. This is where you're going to see the use of interferon, which is produced by infected cells, warning neighboring cells of the viral infection. Phagocytes, upon alert, collect at sites of infection, and basically what they'll do is they will consume invaders. Complement protein activation recruits phagocytes and labels invader cells to be consumed. Inflammatory response results from activation of various sensor systems or tissue damage. Cells of blood vessels allow for fluid transfer into the tissue. This is where you're going to have the endothelial cells open up spaces, and they're going to become basically leaky. They're going to transport more liquid into the tissue. This results in not only the transfer of fluid into the tissue, but also complement proteins and other proteins into the infected tissue. This is going to basically allow for phagocyte recruitment into the tissue damaged area. Finally, fever. Now, it's interesting because fever we all think is a bad thing, but fever elevates the body temperature, and this interferes with the metabolic activity of microbes and enhances innate responses to infection. By the way, think about it from another perspective. As the body temperature goes up, many microbes, their proteins, lose their efficiency, they become basically denatured. Okay? Now, I do encourage you to review the micro assessment of 14.1. Now, if we talk about first-line defenses first, if you were to look at the body and describe it basically, the body's borders serve as the first line of defense against pathogens. You notice there in the pink mucous membranes. The skin is tan. And you notice there that there's some areas that are the boundaries, and there's also a passageway going all the way through the digestive tract, from mouth through anus. And then you'll also notice some of the other mucous membrane areas from the eye, the respiratory tract, the urogenital tract. Now, when we talk about physical barriers, this is basically consisting of the skin and the mucous membranes. You have to keep in mind that the cells of the epidermis are packed tightly together. They rest on the membrane covering of dermis. Now remember, dermis is more connective tissue, if you remember your days of anatomy and physiology. The outermost layer of skin is basically dead cells filled with keratin, which is a water-repelling protein. Therefore, your skin is going to be mostly a dry environment. The mucous membranes, though, line the digestive, respiratory, genital, urinary tract. The mucous membranes wash microbes from the surface. Also, the secretions further help in microbe removal. The mucus from the digestive and respiratory wash the microbes from the surface, and of course you have respiratory cilia which sweep the microbes in the mucus layer, such as in the trachea, up and out. Now this is known as the mucociliary escalator. Where did they go? Well, basically we either spit this out or we swallow it. If we swallow it, those organisms with the mucus get carried down to the stomach, where the acids in the stomach and the rapid changes in the pH for the rest of the digestive system will basically destroy the microbes. Also, urinary flushing. The low pH of urine basically in the genital urinary system help expel many microbes. So we've dealt with those physical barriers. They're kind of a physical chemical barrier. But we've got to go to the antimicrobial substances that are present. One, we said that the skin is kind of a dry membrane, but it also has a salty residue, and that makes it hostile to all but salt-tolerant microbes. We also have the enzyme lysozyme, and this degrades peptidoglycan of bacterial walls that's found in tears, saliva, mucus, as well as tissue fluids, phagocytic cells, and blood. We also have peroxidases, which form an antimicrobial compounds from hydrogen peroxide. This is located in the saliva, milk, body tissues, and phagocytes. Note, catalase-positive microbes can neutralize the hydrogen peroxide. That's why it's important as a diagnostic test to determine if that particular bacteria of strain, that microbe, has catalase. Lactoferritin. Now, this is an iron-binding protein. It's found in milk, saliva, mucus, and some types of phagocytes. And basically what it does is it binds iron. When you bind iron, you inhibit it or keep it away from microbes, which inhibits their growth. And finally, we get into the antimicrobial peptides. These are really interesting because we found them in all sorts of different species. These are usually short chains of amino acids, 15 to 20 amino acids long, and they can insert into the microbe defense, the microbe membranes, and they form holes in the membrane, resulting in the microbe death. For example, one group of AMPs are called defensins, and they have found a variety of them out there. There are actually human ones too. Vitamin D plays a role in the regulation of defensin expression. Also, there are others that are getting attention as perhaps surface pharmaceuticals. Meganins were found in the African clawed frog. Cecropins were found in, of all things, the cecropian moth. But these all do the same thing, and that's neat because you're looking at new ways to provide products that may help fight off infection. Then, of course, we've got the normal microbiota. And basically, I've got to go through first here the skin. Excuse me. You can see the epidermal layer and, of course, comparison with the microbe layer. Now, when we get to the normal microbiota, that's interesting because this is a population of microorganisms. They routinely grow on body surfaces of healthy tissues. Now, look, you got to keep in mind not just the gut, but on the surface of the skin, up in the nasal cavity, etc. There's a variety of them. Now, how do they help in the defense? Well, one of the strategies is to outcompete the pathogens for habitat on the body. This is competitive exclusion, usually by competing for nutrients or binding sites on the surface. Another is that the microbiota releases toxins or metabolites, such as lactic acid, which inhibit the growth of pathogens. The lactobacilli that are in the vagina help keep a low pH, and when they are destroyed by antibiotics, that's when individuals complain about having yeast infections, for example. Okay? Imbalance or disruption of the normal microbiota, as I mentioned, can create conditions favorable for pathogenic growth. Another example is Clostridium difficile, which is causing antibiotic-associated diarrhea and pseudomembranous colitis. And I mentioned also earlier the Candida albicans in the vagina resulting in vulvovaginitis. Now, you have to keep in mind that normal microbiota is necessary for the normal development of the immune system. Some would speculate it kind of primes it and keeps it moving, keeps it active and prepped. Now, for the cells of the immune system, and I turn your attention to figure 14.5, what you see there is a whole host of them. Some of the cells of the immune system are going to play a dual role in both the innate and the adaptive immune system. So we're going to hear from them both in this chapter and in chapter 15. Cells of the immune system travel through the circulatory system. Some reside in the blood, others take up residence in various tissues. Some will also travel through the lymphatic system. Hematopoiesis is the formation and development of blood cells. Now here we've been focusing on the white cells, the leukocytes. We are not going to be talking much about the red cells, erythrocytes, or the platelets, thrombocytes. That I might refer you over to review in an anatomy and physiology text. If you look at table 14.1, what do you see? First off, we're going to deal with granulocytes, and they contain cytoplasmic granules that are biologically active. They usually contain some type of enzyme. And they're identified based on the staining of the granules. You have neutrophils, basophils, and eosinophils. The neutrophils will engulf and destroy bacteria and other substances. They're also referred to as polymorphonuclear neutrophilic leukocytes, which is a mouthful, I grant you. But what you have to understand is this. These normally account for about half of the circulating white blood cells. Now, one of the more recent papers I've read, what will happen with what is referred to as PNMs or polymorphic or PMNs, polymorphic nuclear neutrophils, is some of them, if you notice, they have this kind of lobule sort of nucleus. They will literally form a network or a mesh around a collection of bacteria. And that mesh is composed of their own DNA. So the cells will begin to disintegrate in an attempt to contain and wall off the infection. Basophils are involved in allergic reactions and inflammation. Now, the interesting thing with basophils is they will stain blue or purplish blue if they're given a basic dye known as methylene blue. The granules release histamine and other chemicals to increase capillary permeability during inflammation. So in essence, when you're going to start having this swelling, etc., it's the histamine and the other chemicals released by basophils that are playing the role. And this is interesting because basophils are located in the blood only. Mast cells appear similar in appearance and function. They reside in the tissues. They are not derived from basophils, and they have a different cell precursor. Now, the other one that's very interesting, and I find this interesting because of the fact that if you see a large upswing in the number of these particular granulocytes, you're pretty certain you've got some type of parasitic infection. Eosinophils, and the granules will appear reddish due to the eosin stain, which is an acidic dye. Eosinophils are extremely important in the removal of parasitic worms. They're also involved in some allergic reactions. Usually the granules will contain antimicrobial substances and the enzyme histaminase, which will break down histamine. So they act in some ways to contain an allergic reaction triggered by histamine, but they will also play a key role in fighting off things like hookworms and tapeworms and other things like that. The next one, of course, we're going to deal with is polynuclear phagocytes. Now, polynuclear phagocytes, they make up the polynuclear phagocyte system, which we'll get into in a second. They include monocytes and macrophages. Now, monocytes remain in the blood, but those cells that leave tissues will develop into macrophages with specialized properties. Macrophages are abundant in liver, spleen, lymph nodes, lungs, and the peritoneal cavity. They are an important sentinel cell. Okay, and you can see this distribution here as we talk about the mononuclear phagocyte system. And let me move into another area that's extremely important, and that's dendritic cells. They are an important sentinel cell. They engulf material in tissues, bring the material to cells of the adaptive immune system for inspection. Basically, they help say, look, here is an antigen, this is important, this is an invader. Most dendritic cells will eventually develop into monocytes. And finally, we get lymphocytes, and these are the cells that are responsible for adaptive immunity. Mostly the major groups for these are B cells and T cells, and they are key to antigen recognition. There are some called the innate lymphoid cells, ILCs. They differ from B and T cells because they lack antigen recognition. These types are usually found in mucous membranes, and there is also one type that's called the NK cells, natural killer cells. Okay, moving forward from here, we've got to talk about cell communication, because how do you trigger or round up the troops or have the cavalry come over the hill when the time is necessary? Bear with me with the analogies. Immune cells have to communicate with each other to coordinate the attacks to microbial invasions, as well as to know when to stop. If you don't know when to stop, you will eventually start causing tissue damage to the self. Okay? We'll get more into that later. The process is done via surface receptors, cytokines, and adhesion molecules. And you can see some of this when we talk about cytokines here. But let me first deal with surface receptors. These are integral proteins, so they span the cytoplasm of the cell to provide a change inside the cell. Now, a ligand is a molecule that specifically binds to a select receptor. Once the ligand binds to the cell receptor, a specific change occurs inside of the cell. For example, chemotaxis. Okay? Cells may adjust the receptor types and numbers depending on the immediate situation outside or inside of the cell. So there is some flexibility here. Cytokines, as you see in that table 14.2, are present in very low concentrations. There's a variety of them. These are chemicals used for cell-to-cell communication, may induce local, regional, or systemic effects. You might have heard of the term cytokine storm when someone has pneumonia infection or influenza and their lungs are filling up with fluid and everything else. We'll get into that in a little bit. The binding of the cytokines may induce in the cell growth, differentiating, or even cell death. Yes, even apoptosis. Chemokines, these are triggers chemotaxis in immune cells. Cells then migrate to the location of the need, such as where the inflammation is. There's another group called colony-stimulating factors, CSFs. These are important in the multiplication and differentiation of leukocytes. They can be used to convert immature cells into mature cells during infections and inflammation. There actually have been some across the market now, CSFs that are used to treat patients who've had depressed immunity either because of radiation damage or in an attempt to fight off certain cancers. Interferons were discovered for their antiviral effects, and they have effector action on virally infected cells. Interleukins are produced by leukocytes, and they play a role in innate and adaptive immunity. Tumor necrosis factor. This was discovered due to the role in killing tumor cells. They help to trigger inflammatory response and apoptosis, programmed cell death. They were actually also co-named cachexins because they seem to have an effect that might suppress certain appetite, etc. And they found that they were basically the same thing. When people are fighting off tumors, cancers, TNF levels increase, but it also causes a lot of the patients to sometimes suppress their eating, and so they have to deal with that. Pro-inflammatory cytokines. These are groups of cytokines that act together to generate a response. Now, moving on from here, there's a lot of them, but I don't want you to lose the forest from the trees. We also have forgotten about adhesion molecules. These are basically proteins that allow cells to attach or bind to each other. They play a role in the phagocytes attaching to endothelial cells of the blood vessels and roll and exit the blood. Okay? This is called dipodesis, and we'll talk about it later on, but basically how you would have the white blood cells sort of attach to the wall of the endothelial cells in the blood vessels, kind of stop so that they could eventually roll and eventually get out of the tissue or into the tissue fluid to fight the infection, since it's outside of the blood vessel itself. I also encourage you to review microassessment. And now we're going to get into another cluster of very important areas. This one is called pattern recognition receptors. Now, these are sensors that allow the body cells to sense signs of microbial invasion. They're usually located on the body surface, but they're anchored in the cytoplasm. PRRs help in the first stages of detection of microbial invasion for the innate immunity, and thereby they help shape the immune response to the agent. Now, there are a couple of other terms here. You need to be aware of them because I just got back from a conference recently, and this is another approach to fighting infections. And what they have to do is understand what are these patterns that trigger the responses and how they can help take advantage of them to start to fight against the infection, especially if the infection is antibiotic resistant. Microbe-associated molecular patterns, MAMs, respond to the components of different microbes. This may be cell wall components such as the peptidoglycan, the teichoic acid, the lipopolysaccharide, the piloproteins, the flagellin subunit, microbial nucleic acid. Basically, they reflect that they originate from the microbes. all of these, if I want to call them MAMs. Then you have the pathogen-associated molecular patterns, PAMs, PUBs. I know it's a mouthful. They're similar to MAMs, but they only apply to pathogens. Okay? Then we have the danger-associated molecular patterns, DAMs, or DAMPs. And these are molecules that indicate host cell damage. They're located in 3 distinct areas: on the cell surface, in endosomes and phagosomes, and freely floating in the cytoplasm of the cell. Now, what about PRRs? They monitor cell surroundings. And the best known are the toll-like receptors. Now, before you start thinking toll like toll booth, that's not exactly where they got the name. Toll comes from a German term, so it's like, we see this now, when they first started isolating them. The receptors that extend outside of the cell cytoplasm, they're basically used to detect evidence of microbial invasion by detecting components of the outer layers of the bacteria. So it's sort of like the blind man coming up and feeling, it's an elephant. Now, how do you know it's an elephant? Well, it's got a trunk. I'm feeling the trunk there. I'm feeling the big wiggly ears. I'm feeling the big wall. Now, usually all the blind men they feel one piece and go, it's a long, stringy thing that it must be like a snake, and really what he's doing is feeling the tail. Or it's big tube-like, and it's because he's feeling the trunk. What you have is a bunch of different sensors that compile all of the information. Okay? Trunk, wall, ear, everything, and then you say, it is an elephant. Okay? So they're going to pick up on the outer layers, such as the lipopolysaccharides, lipoproteins, flagellin. There's also one other important subgroup of PRRs, and this is the C-type lectin receptors, which bind to lectins, a type of carbohydrate, but function in dendritic cells to pass information onto the cells of the adaptive immune system. Now, PRRs monitor material ingested by the cell as well. So, these receptors are found in the lumen-facing side of the phagocytome and the endosomal vesicle membranes, and they recognize select features of the microbial nucleic acids. You can see some of that right there in the lower part of the image. They're able to detect double-stranded RNA, select microbial DNA sequences, etc. And it's interesting, they will not become active until a lysosome fuses with the vesicle. Okay? So when that lysosome, remember, when lysosomes are fusing with the endosome, they're going to start digesting. And so now the bacteria cells may be burst, and they may have their inner contents exposed, and that's when you're going to detect single-stranded RNA, bacterial DNA, bacterial double-stranded RNA, etc., etc., etc. Okay? Okay. PR also monitor the cell cytoplasm. Okay? Now, this is where we get into parts where the receptors are found in the lumen, facing side of the phagosomal, sorry about that. PRRs monitor a cell's cytoplasm. There are receptors monitoring the cell cytoplasm, and here's the deal. There are several types. The Rig-like receptor, the NOD, N-O-D-like receptor. Rig-like receptors are cytoplasmic proteins that detect viral RNA. Viral RNA differs from cellular DNA in that it's usually double-stranded, and it lacks the 5' caps that you see by cellular RNAs that possess that. NOD-like receptors can differentiate in a similar way. They're cytoplasmic proteins that either detect microbial components like peptidoglycan, flagellin, components of the bacterial secretion system, or signs of cell damage. Now, these are found in macrophages and dendritic cells. When certain NRLs, in this case NOD-like receptors, in the macrophages and dendrite cells detect invasion, they combine to form a cytoplasmic structure called an inflammasome. This will trigger inflammation by activating the pro-inflammatory cytokines. So you've got this really neat system here. And here you can see it right here kind of floating around, the RLRs and the NLRs. And they're all set to particular components that are unique, more for bacteria cells, etc., than for what you would commonly see in a eukaryotic cell. Okay? Now, the outcome of the pattern recognition is the interferon response. Okay? When PRRs detect viral RNA, the cell responds by releasing interferon. And that induces nearby cells to defend against a viral invasion. The neighboring cells will express inactive suicide proteins. That would be protein kinase R, RNase L, and others. In other words, these are proteins set to destroy viruses if they show up. That's why they're antiviral proteins. Now, IAVPs, the antiviral proteins, okay, can be activated by viral double-stranded RNA. This is a signal of viral infection in the cell. So in other words, think about it this way. You have an infected cell, it broadcasts the signal using interferon to the neighboring cells. I may be infected. Now, all the neighboring cells are set themselves up on alert. They have inactive but have increased their suicide proteins. If they get infected, these proteins will begin to degrade messenger RNA. They will stop protein synthesis, and they will eventually lead to the apoptosis of the cell. Remember, viruses need cells to reproduce more viruses. So what they basically do is set themselves up that if they do get infected by the virus will not be able to reproduce. They will destroy the cell in the process to save the rest of the surrounding tissue beyond that. Non-infected but antivirally alerted cells remain on alert in case of subsequent viral infections. So they stay on alert for a while. Now we're going to talk about the complement system. Now, I've taught the complement system a few times before, and one of the things to keep in mind is this. You're going to have a series of proteins, and I'll let you know what you need to know. Don't panic about all the numbers. If you remember how the clotting routine, the clotting cascade occurred, this is somewhat similar, but the cascade seems to work via all of these complement proteins. The complement system is a series of proteins that circulate in the blood and interstitial fluid. Circulating proteins remain in an inactive form until a microbial invasion occurs. This starts a cascade of reactions that activate the whole system. The activated proteins trigger specialized functions of host defenses to remove and destroy the microbial invasion. The system complements the function of the antibody system, but remember now the antibody is the adaptive immunity system. This is part of the innate immunity system. All complement system proteins start with a capital C. Then they're numbered by the order of discovery that occurred. This is not the order in which they react in the system, so don't get that confused. When the protein is split, the fragments are distinguished by a lowercase letter. So in some reaction step, you may take C3 and break it into two components, C3A, C3B. Complement system activation. There are three different pathways that can activate the complement system. All of them converge on C3 convertase, which splits C3, resulting in activation of the additional steps. You can see these up here at the top. The alternative pathway, the lectin pathway, and the classical pathway. Let's take them one at a time. The alternative pathway, it was not discovered first, but easily activated and triggered. It's an early warning of microbial invasion. It's triggered when C3b binds to a foreign cell surface. The lectin pathway, activated by PPRs called mannose-binding lectins. This is a carbohydrate commonly found in the surfaces of some fungi and bacteria. Once MBL attaches to the surface component, it interacts with other complement system components to form the C3 convertase. Finally, the classic pathway. When antibodies bind to an antigen, they form the antibody-antigen complex, otherwise known as the immune complex. They interact with the same complement system to form C3 convertase. Now, this brings us up to a question. What are the effector functions of the complement system? You may see some of this where the membrane attacked complexes are formed, but let me go back here one slide, and we're going to talk about what happens. When you have the activation of the complement system, this leads to three major protective outcomes. Number one, opsonization. The material prepared for opsonization, prepared basically for eating by some of the other cells, macrophage, etc., they're basically coating the object. Think of it as the microbe now has flags that come and say, okay, this is one of them, it's a bad guy, destroy, eat me up. They're coated with opsons. Now, opsons include C3b. This process makes the microbe easier for phagocytes to engulf. The next response is inflammatory response. The complement component C5a attaches phagocytes to the site of complement activation. Complement components C3a and C5a induce changes to endothelial cells to increase vascular permeability associated with inflammation. So basically, they end up causing spacing between endothelial cells. And this is going to allow or induce mast cells to release selective pro-inflammatory cytokines. You're going to basically, remember now, inflammation is also fluid coming in. So you're going to do some changes to the endothelial cells, and that's going to cause vascular permeability, i.e., liquid moving from blood into the tissues. Finally, the third step is the lysis of the foreign cells. And you see this clearly done on this next photo here. What you have is basically that complexes of the complement system proteins, C5b, C6, C7, C8, C9, they're going to spontaneously assemble in the microbe membranes to form what is called a membrane attack complex, MAX. Now, you notice I throw these in certain bold print on the notes. That means, yeah, you really need to know this. MAX create pores in the cell, and it causes them to lyse. MAX will have little effect on gram-positive cells due to the thick peptidoglycan layer, but they will damage both the outer and cytoplasmic membranes of gram-negative bacteria. Okay? All right, now, moving on, the regulation of the complement system. If you see this, we find that many systems exist to prevent host cell activation of complement systems to shut down activated complement systems. Basically what I'm saying here is there are a bunch of systems that prevent a runaway immune defense. One case, molecules in host cell membranes bind to regulatory proteins that quickly inactivate C3b. Some pathogens have been found to have mechanisms to hijack the host's protective mechanisms. And we'll deal with that later when we get into Chapter 16. And I would encourage you to review the microassessment here. And the next thing that we're going to do is look at phagocytosis. Now, everybody remembers that this is kind of like phagocytosis, phago to eat. If you remember this in A and P, you basically have the cell membranes wrap around and whoop, bring it inside. Normally you engulf and digest the matter, and that includes bacteria cells. That's what phagocytes do. Now, if the infection is not controlled by the resident macrophages, then the macrophages produce cytokines to recruit additional phagocytes, and that includes also neutrophils. The process of phagocytosis, and I would encourage you to review the steps on figure 1414, as you see here. Many pathogens have evolved the ability to avoid one or more of the following steps. This is where we're going to get into host-pathogen interactions and the host defenses, because it's key that you understand what are the mechanisms and how are the pathogens able to avoid them. So let's go through them. First, chemotaxis. phagocytic cells are attracted to the infection site by chemoattractants. These include cytokines, complement proteins, phospholipid release by the damaged host cells. Recognition and attachment. Phagocytic cells have a receptor to bind to invading cells, including those cells that have been opsonized. Opsin kind of helps them to zero in. Engulfment. Once a microbe is attached to the phagocyte begins to engulf it into a phagosome. If it cannot engulf the invader due to the large size, then the phagocyte will release its toxic contents as a means to destroy it. Okay. What about phagosome maturation and phagolysosome formation? Remember that you have a phagosome and it becomes a phagolysosome when basically the lysosomes fuse with that phagosome. The lysosomes are going to bring their digestive enzymes in. So, the phagosome must be activated by a drop in the pH, the activation of the phagosome's TLRs, and the eventual fusion of the phagosome with the lysosome, and the lysosome's degradative enzymes. Okay? Sorry about the typos there. And this forms a phagolysosome. Now, destruction and digestion. Various enzymes are going to act on the peptidoglycans of the bacteria as well as a respiratory burst. This is an oxygen increase, creating reactive oxygen species. ROSs are usually, we're talking about, they're either H2O2 negative or O2 negative or something to that effect, where they would be highly reactive and tend to then react, destroy, or distort proteins or anything else they can get their hands on. It could be lipid membranes, it could be DNA, etc. You also will get the formation of nitric oxide to form reactive agents such along with the ROSs. And finally, belch, no, that's the cell saying that. Exocytosis. After it's digested, the bacteria, the phagolysosome will fuse with the cell membrane to expel undigested debris. Could be cytoskeleton elements, it could be crystals, it could be whatever. Now keep in mind, we have pathogens that have developed strategies against this. That's why knowing this is important. What about the characteristics of macrophages? Well, macrophages, and we'll get eventually to neutrophils. The macrophage's lifespan is months or longer. They maintain a lifespan by regenerating lysosomes. They're present in every tissue. They monitor for invasion, but also engulf dead and dying tissue. Monocytes can differentiate into macrophages to replace macrophages. Monocytes will also migrate to sites of infection, that is, the sites of invasion, and tissue damage. Now, if a macrophage itself is infected, it will form an inflammasome and trigger a strong inflammatory response. Activated macrophages, activated by the pro-inflammatory cytokines from surrounding cells, these cells have a greater killing power and maintain an inflammatory response. What about giant cells? Now, if activated macrophages fail, the cells will fuse together to form giant cells. Very large multinucleated cell found in a chronic inflammatory response. These are found in granulomas. You will see these in cases of, for example, tuberculosis. You will also find giant cells also in cases where there's been HIV infection. And so the cells all form these large clusters trying to fight off. Granulomas include macrophages, giant cells, and T cells. They'll form a concentrated group that will attempt to wall off and retain the pathogenic organisms and other debris that cannot be destroyed. This may interfere in the normal function of the host tissue. For example, tuberculosis, and you'll see this later on. Casein nervosa, basically these blobs are basically contained. If the immune system of the patient begins to falter, or the health falters, sometimes that's when the tuberculosis will break out of these isolated granulomas and then spread into the surrounding tissues. Characteristics of neutrophils. They have more killing power than macrophages. They play a critical role of early defense once they cross blood into infected tissues, but they have a short lifespan in tissues, usually days. Once the granules are used up, the neutrophil dies. They release from the granules destructive enzymes and antimicrobial peptides. We have what has been known as neutrophil extracellular traps, NETs. This is where I was talking with you before about polymorphic neutrophils. Excuse me, I get these. This is where I was talking about before. You get a neutrophil that's really ramped up, and it will discharge and form an extracellular trap, and it's released DNA from the neutrophil. It will function to snare and trap pathogenic microbes, while the granule contents will kill them. Okie dokie. Now, let's get into the inflammatory response. I would turn your attention to 1415. Now, there are 4, actually some people say 5, cardinal signs of inflammation: swelling, redness, heat, pain. The 5th, that's been sometimes included, is the loss of function. So let's go over this, and you can see the injury, the bacteria entering the wound from the injury, and the subsequent activity. This looks a little bit familiar if you remember the clotting response that you learned in anatomy and physiology. Okay? The factors that trigger the inflammatory response, the PRRs play a role in triggering inflammation. Once triggered, the host cells release inflammatory mediators. These are the pro-inflammatory cytokines and chemicals like histamine. Inducers of the inflammatory process include, for microbes, the PRRs and the macrophages detect MMPs. They release inflammatory mediators. Tumor necrosis factor, for example, which acts on the liver to produce proteins to facilitate phagocytosis and complement activation. Also, microbes will trigger complement activation that leads to inflammatory response. Then we have tissue damage, basically DAMPs. That's evidence of damage to the tissue. This will cause cells to release inflammatory mediators. 2 cascades are activated from these mediators: coagulase cascade, which is the clotting cascade, as well as blood vessel permeability. This is where fluid transport will increase. Preparation for diapedesis of more phagocytes. If you take a look at the lower part of the diagram, when the inflammatory mediators are ramping up, they are going to create these I would almost want to call it like a landing zone, or signs. And what happens is you have the loose tumbling of the cells. They'll start attaching and rolling along on the surface of the endothelial cells. And then by diapedesis, they'll squeeze through the enlarged gaps and get into the tissues to fight the infection. Okay. So now we've talked about this, and we've got to also mention a few other aspects of the inflammatory response. The sequence of dilation of small blood vessels, the leakage of fluids from these vessels, and the migration of leukocytes from the bloodstream into the tissues. Some pro-inflammatory cytokines cause endothelial cells to produce adhesion molecules. And we just talked about all of that. That's how we're able to have phagocytic cells roll, tumble, and squeeze between endothelial cells. And this is all part of diapedesis. First, you're going to note that neutrophils will arrive first, and then they're going to be followed by monocytes and then lymphocytes. Clotting factors will wall off the invasion. Usually they're going to stop the blood loss and prevent entry of other pathogens. Then you've got the issue of acute inflammation. This is a short-term response. It's marked by the prevalence of neutrophils. Once infection is brought under control, inflammation decreases with neutrophils removing dead cells and debris. Okay? Chronic inflammation, that's long-term. That may last for years. If infection cannot be kept in control, then what happens is you start facing the buildup of macrophages and giant cells. You may also see the formation of granulomas. Keep in mind, chronic inflammation is usually very common when you have these really tough infections, such as tuberculosis. Okay? Damaging effects of inflammation. Damage from the phagocyte cells, the enzymes and toxic products, can cause tissue damage. No brainer. If the tissues are delicate, for example, membranes of the brain and spinal cord, such as the meninges. The damage can be severe or life-threatening. You also have this group of echinocyoids, the prostaglandins, and these are a group of inflammatory mediators. Remember, echinocyoids are a type of lipid molecule. So how do we reduce this? Yep, NSAIDs, non-steroidal anti-inflammatory drugs. So that's why we take aspirin and ibuprofen to reduce the inflammation. Because what these drugs do, this class of drugs, is they block enzymes that make prostaglandins, and hence they reduce the inflammation at the damaged site. Now, moving on from there, we start coming up with some of the other critical points and kind of wrap this up for this chapter. Cell death and inflammatory response. During infection, cells may die to sacrifice themselves to prevent the spread of the infection. We will have a term called necrosis, that's cell death. Apoptosis is programmed cell death that does not trigger an inflammatory response. Pyroptosis is programmed cell death that triggers an inflammatory response. For example, this process will occur in infected macrophages so as to prevent the cell from being a host cell to the invading microbe. Sometimes you will get these cells to self-destruct rather than allow themselves to be basically a culture medium. Now remember, we talked about some bacteria that are obligate intracellular parasites. That's bacterial. We have some viruses that do the same thing, and we have some other parasitic organisms that will try to invade and stay inside of cells. And the only way to stop them, in some cases, is self-destruction. Finally, we come up with the concept fever. This results when macrophages release pro-inflammatory cytokines. The process will inhibit growth of many pathogens and enhances the rate of various body defenses. Now, pyrogens are substances that are fever-inducing. Endogenous pyrogens include cytokines produced by macrophages. Exogenous, outside the body, pyrogens are microbial products such as lipopolysaccharides. Okay? These will trigger the responses. And with that, we have completed Chapter 14. I encourage you to review the chapter summary, as well as we're going to start moving into the adaptive immune response. These are all key and important areas, so I really encourage you to take the time to review them, read them. You will want to review the key terms as well as the glimpse of history. This gets into basically von Buehring's work and some of his ideas on antitoxins, antisera, and antibodies. Okay? So let's get down to some terms first. Adaptive immunity. This is the immune response protection derived from exposure to antigens. Okay, now look, this is the point. You have to be exposed in one style or another to the antigens, to the pathogen, for you to develop the resistance or to develop a learned response. The response improves with each subsequent exposure, and it usually involves B and T cells. Lymphocyte proliferation is the multiplication of lymphocyte numbers, usually as a response to microbe invaders or foreign material exposure. Therefore, adaptive immunity adapts. The response is tied to the initial exposure to the antigens. It is a learned response by the immune system. You have no exposure, you have no learned response. No presence of a response, humoral or cytotoxic, to the antigen. This is some of the key principles that are going to help us to understand also how vaccines work. Hang in there. We've got a lot to learn. When we talk about adaptive immune response, we've got to get used to some terms here first. First, primary response. The first adaptive response to a particular antigen. Henceforth, the immune system remembers the exposure and is primed for a stronger response upon further exposures. The secondary response, this is a stronger antigen-specific immune response. When we talk about a humoral immunity, humoral refers to fluids, and this is really involving B cells and the antibodies produced by those B cells. They eliminate the microbial invaders and any toxins from the bloodstream or interstitial fluids. The extracellular microbes or antigens are not hidden inside of the host cells. So humoral has a limitation, just as other processes may, if they cannot detect the invader. And sometimes the invader hides by being inside of the cell. Now, B cells develop in the bone marrow. Cell-mediated immunity, CMI, otherwise known as cellular immunity, attacks invaders residing within the host cell. It involves T cells, which mature in the thymus gland. The invaders to the host cell are considered intracellular antigens, which include bacteria, viruses replicating inside of the host cell. This process will also attack cancerous cells. Because of the process of adaptive immune responses, they are heavily regulated to avoid autoimmune reactions. In other words, they are regulated to avoid attacking self-tissues or self-antigens. A naive lymphocyte is one that has not been exposed to any antigen yet, so therefore it will not react to any antigen. An activated lymphocyte is one that is activated by the exposure to the antigen. It will proliferate into millions of progeny, in other words, make copies of itself. The effector lymphocytes are short-lived cells that are used to express traits to help eliminate invaders. Memory lymphocytes are long-lived cells that can be activated quickly upon re-exposure to the antigen. So they're kind of like the sentinels after the initial infection or after the initial exposure. Memory cells play a key role in the rapid secondary response if that invader comes back again. So let's talk a little bit more about humoral immunity. Here we have sort of a regular white blood cell within a blood clot, and you have the fibrin filaments, and this is an EM picture, so you're going to have false color. Now, I mentioned B cells. When naive, that is brand new, B cells detect extracellular antigen. They become activated, proliferate, and differentiate. Some become plasma cells. These are referred to as effector-type lymphocytes. And they produce antibodies against the antigen. Whereas others become memory B cells, otherwise known as memory cells, to become sentinels against subsequent exposure to the antigen later in time. Plasma cells are effector B cells that produce and secrete the Y-shaped proteins we refer to as antibodies. Okay, and you can see that on this diagram. Now, the antibodies are proteins that bind to the surfaces of cells, toxins, viruses, or any other antigen. The antibody has two functional regions. You have a single stem of a Y-shaped molecule, and then you have two identical arms with portions that selectively bind to the antigen. Okay? The antibodies that bind to the antigens will flag the antigen or cell for rapid elimination by other components of the immune system, such as macrophages. The Bcell receptor, and you'll see this on 15.2 here. Basically, this is a cell, at the cell surface of the B cell is a membrane-bound receptor of the same antigen-binding site as the antibody. Once the antigen binds to this B cell receptor, the B cell will multiply, make a lot more copies of itself, and it will produce antibodies. But a second signal is always required. The second signal comes from another cell called a helper T cell in the form of cytokines released from the helper T. Okay, now, cell-mediated immunity. Cell-mediated immunity, we go back one. And let's talk about that briefly for a second. In this area, we deal with T cells. Okay, so B cells are humoral-based. T cells are cell-mediated. T cells are composed of two basic types: cytotoxic T cells, helper T cells. They're distinguished by different types of surface proteins. These are called CDs. You'll hear them referred to as CD this and CD that, usually with a number. These are markers present on the cell surface. Cytotoxic T cells, or CTLs, are responsible for destroying host cells infected with viruses or other cell corruptions. When CTL encounters an infected cell, it instructs the cell to undergo apoptosis, programmed cell death. Helper T cells, TH cells, are responsible for directing various humoral and cell-mediated immunity responses. After native or naive T cell activation, some cells become T helper cells. Okay, so they become the effectors. T cell receptors, TCRs, and again, this is where we go back to there, is a receptor located on the T cell surface that binds to a specific antigen, similar to a B cell receptor, but does not recognize antigens freely floating in the blood or interstitial fluid. The antigen must be presented by the body's own cell surface, for example, with dendritic cells. The attachment will trigger native T cells to mature, differentiate, and proliferate. Some cells will differentiate into CT cells, cytotoxic T cells, an effector form of the T cell. Some activated CTLs will become memory cytotoxic cells, so they will last longer and be vigilant sentinels. Regulatory T cells, formerly called T suppressor cells, have T cell receptors, but stops the immune system from overreacting and responding to harmless substances. So this is again part of the brakes that prevent you from having autoimmune diseases. Now, this is all well and good, but we've got to get a little bit of background here on the anatomy of the lymphatic system, and that I would turn you to figure 15.3. When we talk about the lymphatic system, by definition, this is a collection of tissues and organs that bring populations of B cells and T cells into contact with antigens. Now we have lymphatic vessels, which you see in part here, as well as here. These are vessels that carry lymph, which is a colorless fluid derived from tissue fluid. Now when blood goes from arteriole through capillary to venule, a lot of that fluid that passes from the capillary gets reabsorbed, but some of it is incompletely reabsorbed, and therefore, rather than build up in the tissues, will get picked up by the lymphatic system and carried back eventually to the blood. Okay? But once you have the fluid entering the lymph vessels, other substances may be carried along. These include pathogens, cancer cells, white cells. Also, inflammation may create more tissue fluid that will be also subsequently transported by the lymph. Now, lymph is transported by lymph vessels to lymph nodes and eventually empties back into the circulatory system, as I said, at the junction of the internal jugular and subclavian veins. If we go back from there, we have to also consider the secondary lymph organs. These are sites where the antigens that have entered the body are brought into contact with dense populations of lymphocytes. These are sites where immune cells interact and transfer cytokines. The only sites in the body where adaptive immune responses can occur. Examples of these secondary lymphoid organs include lymph nodes, spleen, and tonsil. Now, the lymph nodes capture materials from the lymphatics, so they basically act as like a filtering site. A weigh station. You'll see a lot of lymph go through them. There'll be lots and lots of B cells and T cells there. The spleen will collect materials from the blood. And then we have another set, interestingly enough, called Peyer's patches. And this is on 15.5 for figures. These are tissues in the intestinal wall that monitor and inspect samples of the intestinal contents. They have specialized intestinal epithelial cells called M cells that transfer materials from the intestinal lumen into the Peyer patches. Also, you have dendritic cells in and near Peyer patches that play a role in grabbing intestinal material and presenting it to the lymphocytes. Now, this is where you get into two types of associated lymphoid tissue. You have MALT, which is the mucosa associated lymphoid tissue. Okay, you'll see that where the Peyer patches are in the intestine, but you'll see it in several other mucosa-related areas. It's a site of critical mucosal immunity that prevents microbes from infecting the body via the mucous membranes. Go back to the earlier chapter. What did we say were the two places where we have barriers. We have the mucous membranes, and then we have the skin. So you have to have primary defenses right there. The skin, otherwise known as SALT, skin-associated lymphoid tissue. These are lymphoid tissues located in the skin that prevent microbial invasions in the skin, in the cutaneous membrane. Now, that's really interesting. And we have these primary lymphoid organs, too. Go back here. You have the bone marrow and the thymus. Keep in mind, bone marrow is where the B cells mature. Thymus is where the T cells mature. Bone marrow is the site also where hemopoietic stem cells reside. So basically you have the stem cells necessary to produce all different types of white cells at the beginning. Maturation occurs later. Of course, you've also got the red cells and the platelets being created in the bone marrow as well. Now, let's move on to antigens. And this is where it gets interesting. What is an antigen? Where did the name come from? It came from antibody generator, antigen. These are compounds that induce antibody production. A more broad description is that any molecule that reacts specifically with an antibody, a T cell receptor, or a B cell receptor. Now look, this does not imply that the molecule can induce an immune reaction. That's what you've got to keep in mind too. We need to get familiarized with a bunch of terms here first, and that is T-dependent antigens. Most antigens require this process whereby the responding B cell requires a signal from the helper T cell as part of the activation process. Usually the antigen has a protein component. But there is also a T-independent antigens. These are antigens that can activate the B cells without helper T cells. These include lipopolysaccharides. Doesn't that sound familiar? Lipopolysaccharides. Yeah, bacteria. Yeah. And molecules with repeating subunits, like some carbohydrates. Repeating subunits. They might have long chains of glucose or lactose or whatever. Antigens, we have to keep in mind, proteins usually induce a strong reaction. Lipids and nucleic acids don't. So when we talk about something being immunogenic, it has a relative ability of an antigen to elicit an immune response. So immunogenic. Small molecules are usually not immunogenic. For example, small antigenic molecules may not trigger immune response in a vaccine. You're getting into vaccines now here. Hence, many smaller molecules are attached to larger molecules to trigger the immune response. That's why you have some vaccines that have adjuvants, or they may be attached. There may be a toxin that's attached to a much larger molecule, just so that the immune system will recognize it and begin to start the immune reaction development. Epitopes. These are antigenic determinants, regions of a molecule the adaptive immune system recognizes. For example, one foreign protein may be 1,000 amino acids in size, but the epitope to generate an adaptive immune response is only 8 to 10 amino acids in size. The surface of a bacteria cell may have a large variety of macromolecules, that is, distinct epitopes, and thus may provide a variety of different epitopes for the development of different antibodies to react to that particular bacteria. Okay? The nature of antibodies. The structure and characteristics of antibody molecules are critical to the function. Keep in mind that antibody binding to specific antigens is as specific as a key and lock interaction. If you take a look at the structure and properties of antibodies, what do you see? Antibodies, otherwise known as immunoglobulins, are Y-shaped proteins with two general parts. You have the Fab, that's the fragment antigen binding. These are two identical arms, and they bind the antigen. Okay? They're at the very tips are where the antigen binding sites are. The fragment crystallized really refers to the stem. And basically, each isolated from enzymatic digestion of the antibodies, you have the antibody monomer, which consists of two copies of long polypeptide chains and 2 copies of shorter polypeptide chains. What holds them all together? Disulfide bonds. Okay? So you can see the heavy chains, the light chains, and what do you see is really at the tips where the antigen binding site is. That's where the variable region. All the rest of it is constant. That's why they have it also FCFAB. Okay? But, because some people, instead of calling it fragment crystallized, they'll call it fragment constant. Okay? But that's not really the truth, as you can see there when you compare constant region with the FC region. The variable region is really part of the antigen binding site. The amino acid sequence of the variable region determines the antigen binding specificity. Antigen antibodies, the binding occurs by non-covalent binding. Now, this is important. We are not forming a permanent bond. It is not covalent, and it can be reversible. They actually use antibodies in a column to purify certain products. When the antibodies bind to the products, they then, after they've run through the solution, they run a second solution that will cause the antibodies to release the product they're looking for. Okay? Now, the constant region includes the entire FC region and part of the FAB region. The amino acid sequence of the constant region determines the class of the molecule. There are 5 types of constant regions. They correspond to the major classes of immunoglobulin molecules. And you will see this in 15.1, right here. And yes, it can get astounding because of the shapes and configurations. But we'll get into that in a few minutes. Let's first go back one step. Because what we have to do is talk about the protective outcomes of antibody antigen binding. First, neutralization. Viruses and toxins must bind to the host cell surfaces to induce damage. Antibodies that bind and coat the virus or toxin block the binding of to the host cell and henceforth neutralize any potential cell damage. So neutralization, just bind to them and keep them from attaching to the host cell. Opsonization. Phagocytes have FC receptors, and hence microbes covered with antibodies will become easier to engulf by the phagocytes. Okay? Think of the antibodies as sort of big flags saying, come on over here and bite on this, eat this, consume this, engulf it. Once the phagocyte receptors bind to the FC part of the antibodies, coating the microbe. Also, complement system activation. Antigen-antibody complexes will trigger the classic pathway of complement system activation, which we talked about in the last chapter. This process leads to the production of opsin C3b, inflammatory response, and the formation of membrane attack complexes. Those are going to punch holes in this microbe cell membrane. We also have immobilization and prevention of adherence. Now, this is a little bit similar, but a little bit different, because binding of the microbe cell flagella or cell pili prevents the bacteria from attaching to the host cell, hence block microbe infection into the host cell. We also have cross-linking. Since antibodies have 2 binding sites, one on each arm, one antibody can basically attach to two different bacteria cells. Okay? This means that multiple antibodies can link to other bacteria and create a matrix, a cross-linking. It looks like basically you get all this clustering up together as you have many antibodies with many bacteria cells. It makes it easier for phagocytes to engulf large complexes of antigen-antibody masses. And finally, antibody-dependent cellular cytotoxicity, ADCC. When multiple IgGs, this is a type of antibody, bind to a virally infected cell or tumor cell, this cell is marked for destruction by natural killer cells. The NK cells attached to the antibodies release perforin or protease-containing granules. Perforin is basically proteins that will puncture through the cell membrane, and basically this triggers the cell to undergo apoptosis. Now, you will note, if you go to table 15.1, know these types, immunoglobulin classes. Now, before you lose sleep and you're looking at table 15.1, I am not asking you to tell me the molecular weight. I am not asking you to tell me all the fine percentages, the half-life in serum. Probably get familiarized with what IgM looks like, IgG looks like, IgA looks like, IgD, and IgE. That's about it. All right? You will get to know a little bit more about this, and I do encourage you that if you ever get interested in immunology and your nursing career takes you into the immunological area, this is going to be a fascinating area to really get familiarized with. But let's start with the basics. All the classes have the same basic monomeric structure, but each class has a different portion of the heavy chain. Some are made up of multiple copies of the basic monomer structure. IgM is the first class produced during primary response to an antigen, the principal class produced in response to some T-independent antigens. It is a pentamer, okay, pent 5. The large site prevents crossing from blood to the tissues. The main role is to control blood infections, the most efficient in triggering the classic pathway of the complement activation system. IgG, the most abundant serum immunoglobulin. It circulates in the blood, but exits vessels into tissues as well, provides the longest term protection of any antibody class. It's transported across the placenta into the fetus's bloodstream, also provides protection post-birth for several months. It's found in colostrum, the first breast milk after birth, and is absorbed in the infant gastrointestinal tract. So this is one of the areas that I mentioned I want you to read. I have an inclusion, an extra little reading for the week, the health benefits of breastfeeding, and this is one of them. We'll get into this more when we get in talk about the next chapter here as part of this lecture for this week. IgAs, most is secreted, is called secretory IgA. It's a dimer, meaning 2 units, and it's important in mucosal immunity, and it's found in mucous membranes that line gastrointestinal and respiratory tracts. Secretions are found in tears, saliva, and breast milk. It's produced by plasma cells in the malt. Most effects are binding, hence effective in neutralization of viruses, toxins, and block bacteria cell entry into cells. It is rarely found in the serum. IgD accounts for less than 1% of serum. Its functions are really a lot less clear. It's believed to be involved in the maturation of antibody response. IgE is very rare in serum. It is tightly bound via the Fc region to basophils and mast cells. It allows cells to detect and react to antigens. IgE-mediated responses are important in the removal of parasitic worms and other parasites. It also plays a critical role in allergies and hypersensitivity. Okay? So that's what I wanted you to walk away from. Now, let's start talking about clonal selection and the expansion of lymphocytes. Clonal selection. This is the process in which the lymphocyte's antigen receptor binding to an antigen, potentially allowing the lymphocyte to proliferate, then differentiate into an effector cell. This process is applied to both B cells and T cells. Lymphocytes that do not bind to an antigen, that is, recognize the antigen, remain inactive. This is how the immune system can avoid mounting a response against self molecules, and hence preventing autoimmune reactions. But what you got to keep in mind here is that the activities of the lymphocytes are altered as they encounter the antigen. Immature lymphocytes, these are lymphocytes that have not fully developed their antigen receptors. Naive lymphocytes, these are lymphocytes that have antigen receptors, but they've not yet encouraged the antigen that they're programmed to respond to. So they're sort of like fully ready to go, but not really have been fighting on the battlefield. Activated lymphocytes, these are lymphocytes that have the correct binding of the antigen to the receptor and have received the second signal confirming that the antigen requires a response, and they are now able to proliferate. Okay? Effector lymphocytes, these are descendants of the activated lymphocytes. They are capable of producing cytokines or other substances. So plasma cells are effector B cells. TC cells are effector cytotoxic T cells. TH, or T helper cells, are effector helper T cells. Memory lymphocytes. These are long-lived descendants of activated lymphocytes. They can become quickly activated if they encounter the antigen again. Memory lymphocytes are responsible for the speed and effectiveness of the secondary response, say after a secondary booster vaccination. Take a look at the chart you see right in front of you. This is the immunoglobulin G levels in the fetus and infant. So what you have to see here is this. Before birth, you're going to have a certain spike in the normal average of adult level of IgG. The infant's going to start picking up. The maternal IgG levels will start going down. Okay, that's interesting. But what we really want to do is start talking about how we're going to have these reactions. And when you have the first exposure to the antigen, your first vaccination, the system gets primed. Why do we have booster shots? So that we can enhance the responses and get a lot more of the effector cells built up and the memory cells built up, so that for an extensive period of time beyond that, you will have immune protection. Okay? Let's talk a little bit more about B-cell response, otherwise known as humoral immunity. This is a very large chart on 1510. Don't panic. And then we're going to move into 1511. What you got to keep in mind is this. The focus is mostly on B cells that required a secondary activation from the T helpers. Antigens that require this are called T-dependent antigens. So let's take a look at this. Naive B cells gather in secondary lymphoid organs. Okay? The B cells take the antigen, this is the T-dependent antigen, by endocytosis. They enclose it in an endosome, break the antigen into fragments, deliver the fragments to the cell surface using MHC class II molecules for inspection and presentation. This is referred to as antigen presentation. This is being presented to the T helper cells. Now, MHC class II molecules, this is part of the major histocompatibility complex. I refer you over to page 401 to review this. And these are molecules on the surface of antigen-presenting cells that present antigens to the T helper cell. So this is a mechanism you will see clearly in certain lymphocytes. The T helper cells also gather in the secondary lymphoid organisms and scan naive B cells. Now remember, when I said naive, it means that they have not been exposed and primed up yet by exposure to antigens. Once the B cells have presented the antigen, the T helper cells will also bind to the antigen and activate the B cell by release of a cytokine. So once they punch their ticket and get their cytokines, the B cell undergoes clonal expansion of that particular B cell. Clonal expansion is saying, I'm making a lot of copies, lots and lots of copies that are specific just for that particular antigen, and exact copies of this particular type of B cell. And, you've got lots of them. Now, if the T cell helper does not recognize the antigen fragment, the B cell becomes anergic, basically is neutral. It consists of a direct induction of peripheral lymphocyte tolerance. That is, it fails. But that's important because in some cases, the B cell, you want to be the most efficient at foreign antigens. And part of the B cell process, where some B cells become anergic, is because they were prepping for self-antigens. This helps reduce the chance of an autoimmune disorder. Characteristics of the primary response. Well, in the first exposure of the antigen, it takes 10 to 14 days for a significant antibody titer to accumulate. As some B cells are dividing, others will begin to differentiate into antibody-secreting plasma cells. Now, by the way, this is just a little pointer. When you have people that suddenly get, like a vaccination or they've been exposed to something, two things to keep in mind. One, if there's a time lag on the first exposure to the antigen, there is going to be a time lag for the immune system to rev up and produce these responses that you need. So you have to keep that in mind. All right? Now we have affinity maturation. With affinity maturation, and here you see the antibodies. With affinity maturation, what are you seeing? This is a form of B cell maturation which favors B cells that bind to the antigen the longest. And they are the B cells that will proliferate. This fine-tunes the binding strength and selectivity of the B cell receptor, and thereby the antibody that it's going to produce that's targeted to the antigen. You're going to have a high sensitivity even at low concentrations of the antigen, which may sound kind of goofy, but think about it this way. You see where the pluses are? Only those that have the best binding to that antigen are the ones that you want to make lots and lots of copies to. Not a partial attachment, not somewhat, it slides off or anything else, because those could be inefficient, and nature doesn't want inefficiency. And they could also inadvertently create some cross-reactivity with antigens that you do not want to be targeting. Okay? Finally, we get into this issue called class switching. Initially, native B cells make IgMs, and once inside lymph nodes, helper T cells release cytokines and trigger the switch into making IgGs. This is going to require a change in the genetic program and deletion of the genes for IgM and IgD. Now, this may sound crazy, but what you really have to think about is you're having some genetic changes occurring in those B cells, so they make the best type of antibody, and that's going to be the IgG. Now, furthermore, some B cells located in MALT, the mucosa-associated lymphoid tissue, will switch over to produce IgAs, and thereby they're going to provide mucosal immunity. Now that we've gone through all of that, we have characteristics of a secondary response. The response is always going to be much stronger. Let's go back here for a second. Response is faster and more effective than the primary response. This is the key factor in the enhanced immune response to secondary exposure to the antigen, such as when you have the string immune response to the booster shot in vaccinations. Think about it this way. If you can see what happens, and in development, knowing this because I used to advise some industries, what they always will try to do is, okay, let's test out the vaccine, and then let's see if we have to give it a booster, and see if we get these massive enhancements in IgG in the secondary response. That's why literally some vaccines you have to give 3 shots. That's why sometimes the vaccines last for 5 years. The first time they were giving out hepatitis B vaccines, you got a shot, and then 30 days later a second shot, and then about another 60 days later, which was 90 days from the first shot, you got a third one. And they thought you were good for 5 years, and then they did subsequent lab studies, and they went, oh no, you're good for the rest of your life. And the only reason you would need a booster is if they asked, you went in and you said, do I need a booster? It's over 5 years, and they would do a titer test. If your titer test showed that you were still producing significant quantities of IgG, you didn't need the booster. And I know this from my own personal life, when I had hepatitis B vaccinations. Okay? Now, what's the key reason for long life of immunity after some initial infections? Memory B and memory T cells monitor for the antigens. They are very sensitive to a small titer of the antigen, and upon activation, the antigen binding, they will quickly differentiate into plasma cells and rapidly produce antibodies, either IgG or IgA. Multiple exposure results in stronger responses. So if you go into a place where you have been vaccinated and you are exposed, but you don't get sick, you're constantly having multiple exposures of these attacks, your immune system is just going to be on sort of this, yellow alert, red alert, off and on. And so you're going to have these stronger responses, and it's going to enhance the memory of that initial vaccination. Now, moving forward from here, what about the response to T-independent antigens? And that's where we go to 1516. Okay, these type of antigens are few, but they're medically important. Lipopolysaccharides, LPSs, and some other molecules are evenly spaced epitopes. These are the antigen-binding sites, and hence, due to the spacing, can bind to many B-cell receptors to the antigen at the same time. As such, B-cell activation can occur without helper Ts. But this effect is not significant to children under 2 years of age. Hence, children under 2 are more susceptible to Streptococcus pneumoniae and Haemophilus influenza bacteria that coat themselves with polysaccharide capsules. Remember, we talked about this a while back. Some of these bacteria that have the capsules, that helps them evade recognition and battles with the immune system. And you can see here this polysaccharide antigen with multiple repeating subunits and the attachments by the B-cell receptors. Now, that being said, we're going to move more into the T-cell response now. This is cell-mediated immunity. And the characteristics of T-cells. Remember, you're going to be talking about cytotoxic T-cells and helper T-cells. And if you notice the CD8, that's the marker that identifies cytotoxic Ts, and the CD4, that's the identifier for helper T. T cells do not produce antibodies, but they induce actions on other cells. Their other cells are the target cells, and they are going to create changes in those target cells. Now, remember we talked about major histocompatibility complex molecules? These are the surface proteins. They consist of 2 types, MHC1, MHC2. So class 1, class 2. And I refer you over to some reference pages if you get a little bit lost here. You can see the antibody, the MHC binding site for antigen, okay? And also on this one, you can see the difference in class 1 and class 2. Class 1 molecules are proteins that cells use to present antigen to cytotoxic T cells. MHC1 presents the antigen made within the cell. In other words, endogenous antigens. All nucleated cells have MHC1 on their cell surface. Okay, great. MHC2 molecules are proteins on the surface of antigen-presenting cells, APCs. Okay? These will present antigens to helper Ts. MHC2 present antigens taken up by the cell, exogenous antigens. Therefore, MHC2 are present only on antigen-presenting cells. These include dendritic cells, B cells, and macrophages. Now, there's an interesting article in the Perspective, and it says, what flavors are your major histocompatibility complex molecules? I would encourage you to review that. MHC at times has been useful in making some tissue typing for the determination of whether tissue transplants or organ transplants will work. But we're going to focus only on the immune characteristics right now. So first off, let's go back one and talk about on 1517, and then we're going to bounce over to 1519. When we talk about the general characteristics of T cells, T cell receptors are present on the surface of the T cell, and it's presenting the antigen to the APC. The antigen is cradled in the MHC molecules of the presenting cell. So you got to keep in mind that both MHC types can present antigens, but T cell receptor recognition is based on recognizing both antigen and the MHC molecule. It's called the whole sandwich peptide antigen, MHC. All right? And here we have an example that we're going to go through this, the antigen recognition by effector Ts. excuse me, cytotoxic T cells and helper T cells recognize different antigens. Cytotoxic T cells recognize antigens presented on MHC1 molecules. If you're an endogenous antigen, such as a viral antigen, okay. If you're an exogenous antigen, this would be something like bacteria antigens, so those outside the cell. Helper Ts recognize antigens presented by MHC2 molecules. That's exogenous antigens. MHC1 is endogenous, okay? When you have the cluster of differentiation, the CD, these are the surface proteins. They are used to identify the different types of T cells, okay? Cytotoxic Ts are mostly going to have CD8s. Helper Ts, mostly CD4. Many cytotoxic Ts are called, in other words, CD8 T cells. Most helper Ts are also called CD4 T cells. CD4 is the marker for HIV to infect cells. That's to help you. You may have heard this in the scientific or the literature in the 80s and 90s. They would talk about CD4 cells, CD4. These are all just the helper Ts. But if you think about it, if NHIV is destroying these cells, they're destroying the mechanisms for both T cell defense and B cell defense, which is why the immune system collapses and the person becomes immunosuppressed. Activation of T cells. Well, here's what we've got. First off, we're going to talk about dendritic cells. These are part of the innate immune system, remember? And they play a critical role in T cell activation. Dendritic cells will reside in skin, mucosa, including the respiratory and gastrointestinal tract. They grab pathogens and break down these cells to produce antigens for presentation to naive T cells in the secondary lymphoid organs. Everybody got that so far? But dendritic cells that detect pathogens produce surface proteins. This is called co-stimulatory proteins, along with presenting the antigens to the pathogens. This triggers the T cell to mature and become primed against those antigens. Those dendrites without co-stimulatory molecules render the native T cell anergic, in other words, unresponsive. And eventually those T cells undergo apoptosis. So there's a way also to prevent, oops, I made a mistake and now I'm starting to cause damage to my liver, kidney, etc., my cell tissue. This creates two situations. It prevents from immune reactions, autoimmune, against your own tissues. So it develops what's called self-tolerance. It also allows for the immune system to develop tolerance to specific antigens. Did you ever think about that? How is it that somebody who's got an allergy and how they try to treat it to get them tolerant to it? Those are some of the mechanisms that are involved in the background. Effector functions of TC, okay, cytotoxic Ts, otherwise known as CD8s, cells. If you take a look here, we have T cytotoxic cells can induce infected cells to undergo apoptosis as well as destroy cancer cells. Now, in healthy cells, the cells release antigens, the self-antigens, that the T cells have been selected against. So they're not going to see them. In virally infected cells, T cells are primed against the antigens that will either release a death package of perforin proteins, remember these are the ones that form pores in the cell membrane to kill the cell, or the T cells will activate the death receptor on the infected cell to trigger apoptosis. That's all important there. Okay? Finally, an activated T cell will release cytokines to enhance the security system of all the local cells. It enhances the antigen detection by the cytotoxic T cells of other nearby cells exhibiting an antigen and activate local macrophages to enhance removal of cell debris and extracellular organisms. Oh, okay. The effector functions of T helper. Remember the T helpers, also known as CD4 cells. Helper Ts coordinate the immune response and hence recognize antigens presented on the MHC2 cells. Now, those are going to be either macrophages or B cells. Once the antigen is recognized, helper Ts release cytokines to activate the B cells or the macrophages. The role of T helper cells in B cell activation. Okay. I want to make sure that we get this. Okay. If a B cell presents an antigen to a T helper cell, and the T helper cell will release cytokines to enhance maturity of the B cell. But in conjugate vaccines, now conjugate vaccines are where you're going to have a combination of something that would be your hapten, let's say a polysaccharide, a lipopolysaccharide, in a protein fragment. That's your antigen. Now the B cell can't react to the hapten. Remember we talked about this? The lipopolysaccharide just is not enough to get a response. So you have to attach it to a bigger molecule. Okay? So once you have that, the T helper reacts to the antigen fragment and will still stimulate and mature the B cell for further immune responses. Now, a hapten is really a substance in which antibodies bind, but it doesn't elicit the production of antibodies unless it's attached to a larger carrier molecule. An example of this is the tetanus toxoid protein. You want to treat someone or prevent them from having tetanus, you have to use a tetanus toxoid, and then you have to attach it to a larger molecule. Okay? That's how you get the individual primed to defend themselves against an attack by tetanus. Now, the role of T helpers in macrophage activation. Okay? I want to make sure that we get this down. Macrophages are going to engulf and degrade invading microbes. Okay, this is phagocytosis in the phagosome. But if the microbes cannot be controlled, the macrophage presents some antigen fragments via MHC2 molecules to the helper T cells, which in turn release cytokines that enhance macrophage activity. The macrophages that are activated demonstrate a ruffled plasma membrane. They have also increased number of lysosomes and produce the antimicrobial substance nitric oxide. If the macrophage response is insufficient, the macrophages and T cells will fuse together, forming the giant cell. We talked about this earlier, but this is worth bringing back again. The giant cells fuse with other macrophages and T cells to form a granuloma. This is going to wall off and isolate the microbes from spreading to other tissues. Okay? This is how, in part, we end up with granulomas from tuberculosis in the lung tissue. Okay? They're basically going to form the granuloma to isolate, wall off the microbes from spreading to other tissues. And this is because you're going to have activated macrophages fuse, and they're going to make this. Now, I'll give you a couple of pages there where you can look at some of the images. We'll later go through TB when we get into those chapters 21. Finally, we have this issue about subsets of T helper cells. The effector T helpers have various subtypes. They are denoted by Th1, Th2, Th17, etc. Each different subtype plays a special role in the immune responses to various intracellular and extracellular pathogens. That's all that you really need to deal with right now. Finally, we're going to be going through NK cells, natural killers. By definition, innate lymphoid cells, which are lymphocytes but lack antigen-specific receptors, like the B cells and T cells. NK activities assist in the adaptive immune responses. NK induces apoptosis in antibody-bound self cells, usually infected with intracellular pathogens such as a virus. This process allows for ADCC, this is antibody-dependent cellular cytotoxicity. NK cells have Fc receptors for IgG antibodies on their surface. NK cells attach to the Ig antibodies bound to the cell and release perforins and protein-containing granules into the cell, which trigger apoptosis to the cell. Okay? Now, this, as we move into 1524, is sort of the big picture here. Okay? Nope, let me go back here. Lymphocyte development. And this is kind of a summary of the adaptive response. I'm going to go over some of the brief points. And then there's a genetic recombination issue here. Don't lose a lot of sleep on it. I'll just cover it briefly, but I want you to be somewhat familiarized with it. The development of B cells and T cells derive from the hemopoietic stem cells. In other words, basically the stem cells that are in the bone marrow. But the maturation of B cells occur in the bone marrow. The T cell maturation occurs in the thymus. The maturation and development of effector function is summarized in figure 1524. You can see it here. How do B cells develop the diversity of binding to 100 million plus different epitopes on different epitopes from all of the diverse pathogens and other substances in the outside world? This is where the question of generation of diversity for B cells and perhaps to a degree T cells. Susumu Tonegawa solved the puzzle which earned him the Nobel Prize in 1987. Here's the key point. You had gene fragment switching in the variable regions of the antibody, and this yields a multitude of combinations. Now, you don't have to memorize all this, just listen carefully to it. What you have to think about is this. If you looked at the DNA of a hemopoietic stem cell, you've got these different regions. Regions that are variable for the heavy chain and regions that would be variable for the antigen binding site. Okay? And we're going to focus on the B cell now, but the process is very similar to the binding site development for T cells. What you have are these sections called variable, diversity, and joining regions on the DNA. And they're different combinations. And what you literally have is a switching, removal, patching together, etc., that creates this myriad of combinations for a number of B cells. Each B cell makes up only one combination for all of the antibodies it produces. So what you want to think about is this. That's going to be a unique binding site composed of V, D, and J genes. Think about it from the analogy that I give you. You have a machine. It spits out a unique key for an epitope. The epitope would be the lock. And what happens is it takes a very long strand of metal, slices out pieces of, let's say, from the V area, the D area, and the J area, mix and matches them, and there you have this unique key. You're going to make a lot of different matches. Some of them are going to be the risk of self, going against self proteins. Those are going to be neutralized by anergy. But other cells are going to produce some of the most incredible combinations. But the other thing that you're going to have to deal with is this. Once you've set the combination, the nucleotides for the genes not used, as I mentioned, are purged out from the genome. But when you start refusing the D and the V and the J, the process may not be completely exact, and hence you're going to have the potential of two B cells making the same combination. Yeah. But you also have the possibility of binding sites having differing binding strengths. You know, one might be really good, one might be really weak. One might hold it really tightly, and one just kind of, gives it like a high five and see you later. That you don't want. You want the binding strength to be very high. So you're going to select for these. The negative selection of self-reactive B cells. How do we keep from making antigen binding sites with antibodies targeted to self? So basically, how do we avoid having massive autoimmune distortions? most of the maturing B cells undergo what's called negative selection, which eliminates any B cells with self-antigen targets. Those that fail negative selection are induced to undergo apoptosis. Sometimes the B cells get by the negative selection and will react to self-antigens. This is what is going to account for some of the autoimmune diseases that we have. Now, we also have a positive and negative selection of self-reactive T cells. This only occurs in the thymus. It's a two-step process for T cells to rule out self-reactive T cells. Step one is a positive selection. T cells must bind to an MHC they recognize to develop further. Those that fail are eliminated. The second step is negative selection. T cells that recognize MHC molecule binding and recognize self-peptides are eliminated. As a result of this strict maturation, over 95% of the developing T cells are destroyed via apoptosis in the thymus. Now, that may seem impressive, but it should be, because that, even though we have a vast diversity of antibodies that can be made to defend us, we also make a vast diversity that are selective and protective. Okay, I encourage you to review the chapter summary. And now what we're going to do is move into chapter 18. I wanted to include this because it's very important. We're going to touch upon a couple of different approaches, applications of the immune system. Okay? Now, obviously, you see someone here and they are setting up an immunoassay. In your lab, you're going to do a simulated immunoassay online, thanks to some of the wonderful people at the Howard Hughes Medical Institute, and that will go through a process and teach you how to do this. But what I'm going to focus in on is immunizations and immunological testing. I do encourage you to review the key terms and the glimpse of history. And you do need to go over some of the terms. Immunization is the process of inducing immunity. Active immunity is immunity as a result of the patient being exposed to the antigen and develop an immune response to the exposure. You need to note that there are long-term effects here. Passive immunity is immunity as a result of antibodies being transferred to the patient to provide a short-term immunity. I encourage you to review the extra handout that I've provided for you in this particular week, entitled Immune Benefits of Breast Milk at a Glance. It's quite surprising the amount of material that mothers give in colostrum, etc., to help the newborns to develop a functional immune system until they get to hand over and take over their immune system. Now, if you take a look at the principles of immunization, we talk about natural active immunity. This is acquired immunity due to exposure to an infectious agent. So you've got smallpox, you got smallpox, you got chickenpox, you got chickenpox, you got measles, you got measles. Then we have artificial activity immunity. This is acquired immunity due to exposure of the antigens of the infectious agent. Example, vaccination. Excuse me. Then we have natural passive immunity. This is acquired immunity due to the transfer of antibodies, immunoglobulins, during transfer from mother's placental milk, placental blood or fetal blood, or due to immunoglobulins present in breast milk given to the patient. Okay, and then we have artificial passive immunity. This is acquired immunity due to immunoglobulins injected into the patient to prevent infection from a pathogen. This might be anti-sera or antitoxins. This might be the serum that occurs for IgG for individuals who might have been exposed to hepatitis A, but basically they get the IgG from individuals who've recovered from hepatitis A. Okay? Antiserum is a preparation of serum containing protective antibodies. Antitoxin is antiserum targeted against toxin of pathogens. For example, diphtheria toxin. Hyperimmune globulins. This is an antiserum prepared from the serum of donors with high amounts of antibodies to a specific disease agent. Example, hepatitis B. Prior to the onset of hepatitis B vaccine, that's what the individuals got, but there was risks for that. And that's why it was favored to finally get a hepatitis B vaccine out onto the market, particularly for healthcare providers. Immunoglobulin, IgG fraction of blood plasma pooled by many donors. The antibody mix and includes plasma from vaccinated patients. This is used to protect unvaccinated patients recently exposed to certain diseases, as well as used for immunosuppressed people who exhibit low levels of antibodies. So let's go into this, and I would turn your attention also to 18.1, 18.2 tables. And you can see the vaccines used to prevent bacterial diseases. And then we have the vaccines used to prevent viral diseases. And there are quite a few of them. Sadly, there have been a few in the process that were recalled because they caused problems. There was a rotavirus that was working well, and a respiratory syncytial that was out there, but unfortunately it was found to cause severe adverse effects and got pulled off the market. Now, when we talk about a vaccine, it is a preparation of a pathogen or its products used to produce active immunity. Keep in mind that vaccines do not prevent infections to the individual, but also protect an infection outbreak to occur in a population. I want to clarify that. Keep in mind that vaccines do prevent infections to the individual, but they also are used to protect an infection outbreak to occur in a population. Now, why I wanted to correct that, I made a goof and I apologize. If you have sufficient number of the population vaccinated, they will act to blunt the outbreak of a particular disease. Now, some vaccines have to be administered in combination, thereby minimizing the number of injections required for patients. That's where you're going to get, for example, the MMR, measles, mumps, rubella. Vaccines should ideally induce both humoral immunity, the production of antibodies to neutralize the bacteria or virus, and cell-mediated immunity, so they create T cells primed to limit the growth of bacterium or virus located inside the host cells. Vaccines should be, ideally, inexpensive, stable with a long shelf life, easy to administer. There are some limitations there, especially in developing countries, when they've tried to move vaccines, and it's hard to keep a cold chain. In other words, the vaccine made had to be in cold refrigeration, and it's hard when you get to some of the subtropic or tropic nations, or there's power outages at the hospital, etc. Sometimes what they'll do is they'll lyophilize the vaccine, so they'll basically freeze dry it. And when it's needed, they will add sterile water to it to activate it and then make it an injectable formulation. Some of the other terms that you need to know about are herd immunity. This is the phenomena that occurs when a critical concentration of immune hosts prevents the spread of an infectious agent. That's what I meant by, if you vaccinate one, it starts to have an effect in the general population of whether there's going to be a spread of a disease. Now, this can occur by having a select high percentage of the population vaccinated against the disease, or a certain high percentage of the population already immune due to previous exposure to the disease. Here's a couple of points. Herd immunity in certain areas. For example, usually it's about 80, 85%, 90% to blunt some of the childhood diseases. The problem in some countries now is they're recognizing that they've gone well below the herd immunity numbers for those against things like measles or mumps. In some cases, they've done surveys and found, wow, we're supposed to be up at about 78% to 80, and they're down to 68. And the other thing is that you start having an influx of migrants. Migrants aren't bad, but if they've not been vaccinated, you start having problems because you add to the population of susceptible individuals for that disease. They have not been vaccinated, and therefore they increase the risk of an outbreak, spreading of the disease, which would affect all of those that are not vaccinated and not protected against the disease. Ring immunization is another concept that you need to know. This is the controlling an outbreak by vaccinating and monitoring a ring of people around each infected individual. This was a strategy that Henderson did to help begin the process of finally eradicating smallpox. Now, here's the point. If you don't have enough vaccination, which is one of the challenges he had, to cover a whole population, what he just turned around and did was have his workers do ring vaccination. You have patient A. Everybody that knew patient A that came in contact with him that had the smallpox was vaccinated. You didn't have to vaccinate the whole village, just all those people that came in contact with patient A. And eventually it reduced the incident. Now, part of the point you have to understand is a little, very important point in the background of smallpox. There is no reservoir. In other words, it's not going to be just found in dogs or bats or pigs. Smallpox only reservoir is human beings. If everybody is isolated and everybody is vaccinated, no smallpox. Just to give you an example. Attenuated vaccines. Now, we've got to get into attenuated versus inactivated. Okay? Attenuated is a vaccine created from a weakened form of the pathogen that cannot cause disease. The weakened form, whether it's bacteria or virus, replicates in the host body and stimulates the immune response, may produce a mild strain of the disease, especially in immunocompromised individuals. This is a danger. When you have an immunocompromised individual, usually the standard precaution is you do not give them an attenuated vaccine because depending on how weak they are, it may instead start a full-blown effect or at least make them mildly sick. And if they're already weakened because they're immunocompromised, you don't want to make it worse. Now, this form of vaccine may be used orally to stimulate IgA, so that's mucosal immunity, especially if the pathogen normally is infected orally and the virus can be shed in feces. Okay? The pros, well, 1 to 2 doses provide long-lasting immunity. The cons, not advised for pregnant women because the strain may cross the placenta. Many vaccines of this type require refrigeration. That means a cold chain for delivery and storage, which is difficult in developing nations. Now, in inactivated vaccines, this is where the vaccine is based on either fragments of the pathogen or the actual killed pathogen. It cannot cause infection or replicate in the host, but the immune response is limited. A lot of times it requires boosters to achieve sufficient immunity. There are different types. The inactivated whole agent contains the killed microorganism or inactivated virus. One of the types that was with polio, they just added formalin. It basically neutralized the proteins that make up the viral coat. Bang, bang, it's a dead virus. You could inject it. Toxoid vaccines, they contain inactivated toxins that are caused by the bacterial pathogens. Subunit vaccines contain key protein fragments or antigenic fragments of the pathogen. And then we have the virus-like particle vaccine. This is basically empty capsids. In other words, the viral particles are there, the proteins. And that would be anyways what you're going to have for the antigens, but they lack the nucleic acid, so they're not going to have any replication. Now, the polysaccharide vaccines, these contain the polysaccharides of the capsules of select bacterial pathogens. They're not effective for young children, sadly. The polysaccharides are T-independent antigens, and therefore they're only given to adults. Conjugate vaccines, these are polysaccharides linked to proteins which convert polysaccharides to T-dependent antigens. Now, an adjuvant. An adjuvant is a substance that enhances the immune response to the antigens. It comes from the French adjuver, to help. They may function by providing danger signals to the dendritic cells and thereby draw the dendritic cells in to pick up the antigens. Some types may cause an intense inflammatory response. The common types of adjuvants are alum, which is an aluminum hydroxide and aluminum phosphate mixture, derivatives of lipid A, squalene, and even some cytokines. Also, adjuvants have been found to act as stabilizing agents for vaccine formulations. Now, I wanted to bring up one thing that was really important, and let's see if I can get that. That's mentioned in later on. An example of a vaccine strategy. This was the campaign to eliminate poliomyelitis, and I would encourage you to take a peek later on page 711. which is in figure 2614. Here's the situation. We had the inactivated polio vaccine, which was the IPV, which was the Salk vaccine. And that consisted of an inactivated viruses of all 3 polio strains. You had a lower disease rate, but required a series of injections. Also, if only using IPV, the wild-type polio virus could still replicate in the intestine and be shed in the feces. So it would end up in the sewage, and if you have poor sewage containment, it could end up in rivers and infect others downstream not vaccinated. The oral polio vaccine, OPV, that's the Sabin vaccine, consisted of an attenuated strains that replicate in the intestinal tract. They're taken orally. The advantage is that they were cheaper to make, induces better mucosal immunity, because you're going to get a secretory IgA response, and provides better herd immunity. The disadvantage was that the attenuated viruses can mutate back to wild type. Again, being disease-causing viruses and becoming virulent and can be shed in the feces. Now, the example that's provided is the Israeli situation. In 2013, wild virus was found in high numbers of the population. High IPV rate in the population existed, but IPV patients were still making wild-type virus in their intestine and shedding it in their feces. In other words, they weren't getting disease, but they were shedding this virus in the feces, which, of course, doesn't mean that it all goes down into some hole in the ground. It can leak into water systems. So this is a risk to the unvaccinated population due to the presence of wild-type virus. This resulted in the distribution of the OSV, the oral type, to the population that only had IPV. Okay. Now, moving onward, importance of childhood immunization. Take a look at the data that you see there. Common childhood diseases and deaths due to these diseases has been drastically reduced. Okay? Now, I'm bringing this up because you are all going to be healthcare providers in one way or another, and guess what? There are people that say, no, I don't want to take the vaccine, etc. That's one. Also, keep in mind one other thing. There are those that want to get, they think that the vaccines are for little kids. So as they get older in the 40s and 50s and 60s, and we'll talk maybe a little bit about this in epidemiology, suddenly they start having an increased risk of tetanus because of their work site. And what it is that tetanus bacteria are very common in the soil, and so they're doing construction or whatever else, they cut themselves. They've not had a tetanus vaccine since they were, 15. They're now 45, and now they're wondering why they're getting tetanus. Okay? Common childhood diseases and deaths have been reduced. Yep. Protected population thinks diseases will never return. Hence, no more need for vaccinations. Wrong. Also, exaggeration and excessive focus on rare adverse effects causes parents and patients to think the risk of vaccine injury is much higher than in reality. That's why I've given you some of those YouTube videos that I strongly encourage you to review. And I really strongly encourage you to view them because you are going to be at the forefront of dealing with patients and parents who will have the debate over the vaccinations. And you need to be aware of some of these issues. Okay? And the problem, after all of this, is that when the immunity, the herd immunity in the population is reduced, we start having outbreaks in childhood diseases. And you can hear this in different parts of the world. Yes, even in places like the United States, the UK, Canada, etc., where there's just a lowering of the herd immunity to the point that there are outbreaks. Measles, mumps, chickenpox, rubella, whooping cough, etc. And it's not nice because the kids and the adults suffer. There is a national vaccine injury compensation program. It's paid for by a small tax on each vaccination, which is a no-fault program compensating vaccine injury claims to help stabilize the U.S. vaccine market. Without that, a lot of the vaccine makers would not take the chances of making the vaccines. What do we have for current progress in immunization? Well, I'm going to tell you this. There is some very interesting developments. One, DNA-based vaccines. And that is, you take segments of naked DNA from an infectious organism, and you're looking for just a couple of antigens, and you inject it directly into the host muscle tissue. The muscle expresses the DNA for only a short period of time. It produces the antigens that you're looking for, creates an immune response, and eventually you have immunity to the pathogen. Edible vaccines. Yep. I'm not crazy on this. This is for real. They've found that they can insert certain genes of antigens for diseases into food crops. And they've been very successful with potatoes. They started initially with tobacco plants only because those were the best and easiest to work with. And now they're moving on with several other food crops, from sweet potatoes and bananas and potatoes, regular potatoes. The idea is this. Look, in developing countries, it would be easier if they consume the antigens and develop the immunity via the food crops. Also, it would be helpful for using plants to develop vaccine antigens for extraction, purification, and the rapid manufacture of new vaccines. Finally, peptide vaccines. And this is basically composed of peptide fragments to key antigenic peptides for the pathogens. These could be stable from heat and contain no extra materials, and hence reduce the chances of side effects. Okay? So from there, we're going to move through the case presentation 18.1 and the microassessment. Now we're going to go into immunological testing. And this is where it gets interesting, because again, this is another aspect of immunity, immunological principles, etc. Immunological testing. This takes advantage of a specificity of the antibody-antigen interactions in diagnosis. Well, it's very simple. Think about this. One clear example. You've all been probably tested. The Mantoux test for tuberculosis infection. That's the Mycobacterium tuberculosis. When the patient is given the PPD, the purified protein derivative of the organism by a skin injection, the patient, if having tuberculosis, would exhibit a redness and a firm swelling at the injection site. If they test positive, the patient... Now, there is one other thing. We go over here. And I refer this to the pages. You'll see this on 554 and 446. Now, if you don't react heavily, then you've never been exposed to tuberculosis and everything's fine. Now, there's one other thing, though. In the United States, they don't use this vaccine. In other countries, they do. It is called the BCG vaccine. Stands for Bacilli Calumet Guerin. It is a modified mycoplasm, mycobacterium, similar to tuberculosis. And it's an attenuated vaccine. If you get it vaccinated, you've got to carry a card around the rest of your life that basically says you were given this vaccine, because you will always test positive on a BCG test. Okay, I should be on the PPD test. What happens is, if you end up positive on the PPD test, then they take you over for a chest x-ray to see if you actually have tuberculosis present. Now, hopefully some of this may be modified with the onset of gene testing and other things like that. But let's move into some of the other principles of immunoassays. Here you see a situation. You have a solution containing known antibodies to Treponema pallidum, and then you have an unknown organism. The antibodies will bind to the bacteria, and that would suggest that you either basically you have the parasite, you have the bacteria. Treponema pallidum is also the organism that causes syphilis. Now, you could also take the serum from the patient, and if the individual's been fighting syphilis, you can take the serum and it'll have the antibodies to it, and you mix it with a known sample of Treponema pallidum, and you're going to get still this cross-reactivity. Excuse me. Now, we have immunoassays. These are in vitro, that means in glass, as opposed to in vivo, which would be in the living organism test, that uses the specificity of the antigen-antibody interactions to detect and quantify the presence of antigens or antibodies in a given sample. Now, we talk about seroconversion. In seroconversion, this is the change in serum that lacks specific antibodies to serum that contains specific antibodies. For example, as infection progresses, the antibody count for a particular pathogen increases, whereas in a previous infection or in a vaccination, the levels of antibodies will remain at a steady state. Okay, that seems like that makes sense. And basically, when we get to, I just want to make sure that I'm in the right area. Okay. The quantifying the agent, the antigen-antibody reactions, we use concentrations of antibodies in a serum sample, and this is usually done by making serial dilutions. And this is where we get into the concept of titer. Titer refers to concentration. So this is expressed as a reciprocal of the last dilution that gives a detectable antigen-antibody reaction. For example, you see all these little wells here on this plate. If a reaction occurs at the dilution of 2 to 256, but not 1 to 256, but not 1 to 512, then the antibody titer is 256. The microtiter plates, and these have been an incredible tool, they replace the larger bulky test tube tests. And each well in the microtiter plates is basically for smaller samples. You can have special equipment for the reagent mixing and analysis, and hence you can get faster results. Okay? Now, how about obtaining known antibodies? Well, if you take a look on perspective 18.1, this is where they're going to talk about some of the concepts of how we get monoclonal antibodies or polyclonal antibodies. Polyclonal antibodies are antibodies made from animals, usually, that bind to a variety of epitopes to a specific target agent, the whole agent or a purified agent. For example, whole killed cell or purified proteins from the cell membrane. Therefore, you're going to get a mix of antibodies that are going to target different parts, that is, different epitopes of the protein structure. But the great thing is that they're all directed against the target agent. Now, in anti-human IgG antibodies, they're commercially available. These are antibodies which bind to any human IgG constant region. And you can get those basically from commercial suppliers. Monoclonal antibodies, otherwise known as MABs, recognize only a single epitope. They're difficult to make, but they've now been able to use these techniques to make batches and batches of them. They're very valuable if they are selective for a known antigen. For example, all right, folks, how many of you know someone that took a pregnancy test? If they did, they probably were looking for a molecule called human chorionic gonadotropin molecule. The HCG test, and you've probably seen those little stick, pee on the stick, etc. HCG is the first protein hormone that gets kicked out by the developing embryo that eventually signals the ovaries to continue to produce estrogen and progesterone so that the uterine lining stays nice and rich and very vascularized, so the developing embryo can basically do what's called nidation, implant itself into the uterine wall. So this is how we are able to get much, much more sensitive tests. HCG will start spilling out. It can be detected in blood. It can be detected in urine. And these tests today are just incredible. The production also of what we call hybridomas. Okay, what's going on here? In the mid-70s, a couple of scientists came together and they said, look, I make a cell that makes monoclonals, but it lasts a couple of cell passages and then dies. And so these cells were obtained from the spleen. And what they tried to do was they mixed those cells, because basically the spleen cells would only have a short, limited lifespan. They mixed them with what is called an immortal cell line, which is basically a cancer cell line, these myeloma cells. And they fused the cells together. And when they fused the cells together, they found that the cells would do what they had been previously expected to do, make monoclonal antibodies. But now they're immortal. These hybridomas, as they're called, the fusions of B cells and myeloma cells, now basically allow you to basically make lots and lots of antibodies for a limited amount of time. Just keep those cells happy and well fed, and they'll be producing them morning, noon, and night for years. And that's how they're just kitchen labs that make this in some places. I remember having talks with some of these researchers. They say, you bring us the B cells that you want, and we'll make up tons of antibodies for you. Now, these monoclonal antibodies have created long-term supply for the detection of drugs, diagnostics, diseases, you name it. And then the other thing was we came up with, why don't we use the antibodies for medications, for basically fighting off types of cancer and things like that. And so medications with MABs, monoclonal antibodies, are used as a drug to provide passive immunity with select types of cancer. And drugs that have the MAB suffix in the drug name are usually containing or made up of monoclonal antibodies. Tagged MABs, that's when you have an antibody and a drug conjugate. And monoclonal antibodies with an attached drug or toxin are used for selective destruction of particular cells, like cancer cells. And then you have recombinant humanized monoclonal antibodies. These are modified antibodies. They're created in animals or animal cell lines that have been modified for humans and yet have extended half-life in patients to destroy cancer or other disease targets. Drugs that have that usually have a Zumab suffix in the drug name. Okay, let's go through some common immunoassays and probably learn some things and start moving toward wrapping up this week. I know this is something that you probably have taken a break or two, and that's okay. But you want to get this down because this is going to help you even with the ELISA lab that you're going to do later on. Okay? Common types of immunoassays. Now, immunoassays that use labeled antibodies, and they label it with a radioisotope, but now that's been replaced with a color-producing enzyme or a fluorescent dye molecule. Nobody wants to have radioisotopes in their labs anymore if they don't have to have them. The advantage of this is that the immunoassay is very sensitive to the target antigen. It's more likely to detect the antigen in question if it's present. Various research and clinical labs use these types of tests. Now, immunoassays that involve visible antigen-antibody aggregates, okay? If you think about the hemagglutination that you saw with the ABO tests, well, we'll get back to that in a few minutes. These are cheaper and now replaced by more sensitive tests that use labeled antibodies. But they still exist, and they are inexpensive and technically simple. As I said, the ABO agglutinin blood test that you may have seen before. Let's talk about immunoassays that use labeled antibodies. The direct immunoassay, which is known to identify an unknown antigen, and you can see this here. You're trying to locate the specific antigen in a clinical specimen. So, what do? What you're going to do is you review 18.5a for the steps. Notice that you have to have a washing step. Why is this necessary? If you put into your well, you're going to have excess antibodies that you have to wash out of there. So basically it's a matter of fill it up, expose it to the antibodies, wash it off by basically removing the fluid and adding the next level of antibodies, washing those out, and then all you have are the different types of antibodies. The direct approach is you have labeled antibodies, you have the unknown antigen, you add the antibodies. If the antibodies and the antigen interact, basically if the antigen's there, those antibodies are going to bind. And fine. If you have excess, you can just wash it off. And then you test for the label. And the test is usually going to be because of what you've added on, that detectable marker. It could be a fluorescent dye molecule, or it could be an enzyme that you add something later, and this is where we're going to get into this later. So whatever it is, it'll be detectable. Now, that's the direct method, okay? The unknown antigen is always going to be attached to that solid surface of the microtiter plate. Let's talk about indirect assays. This is used to detect antibodies of a given specificity in the patient's serum. You have an ELISA that's going to be looking for antibodies to lupus in that lab. Now, they're called indirect because they require a labeled secondary antibody to detect the first primary antibody. So if you look over at section B there, the known antigen is attached to a solid surface, for example, the walls of the microtiter plate. The unknown antibody is attached to the surface antigen. Then you wash them to remove any unbound antibodies. Then the next step is to detect the bound antibodies. Those are the primaries. So you add an anti-human IgG antibody, which will bind to the primary antibodies. Okay? You wash it again to remove any unbound antibodies and then test the label present. If it's present, the primary is present as well. A lot of times now they're using fluorescent dye molecules. Okay? And they are very cheap and they're very easy to attach to the antibodies. Okay? This is where you get to enzyme-linked immunosorbent assay, ELISA. It uses an enzyme-linked antibodies which bind to a target antigen. Now the colorless substrate is converted to a color via an enzyme. Okay? And so what happens there, the enzyme in this case is horseradish peroxidase. And basically what horseradish peroxidase is going to do is it's going to change the color. And that's what you want. And they have many of these diagnostic tests for pregnancy and Lyme disease and West Nile fever and giardiasis and HIV, etc. Now, direct ELISAs that you see in, let's see, 18.8, which I'd bring you over here. You have the sandwich method. Okay? The specific antigens are captured by specific antibodies attached to the cell wall. That's to the well wall, I should say. Then the labeled antibodies are added to identify the presence of the target or captured antigen, as you can see there. Okay? In Western blotting, it is very common what they'll do is they'll have proteins, which are potential antigens, and they're basically spread out, separated by size through a gel. This is called an SDS page gel. And usually it's done by the process of electrophoresis. The separated proteins are then transferred to a nylon membrane, this is called a blot, and then they're exposed to antibodies specific for the target antigen. Then you wash off the unbound antibodies, finally add an anti-human enzyme labeled or radioactively labeled antibody. That second antibody is going to bind to the first antibody. After your wash, the blot is analyzed for the presence of the radioisotope or the enzyme staining that occurs. Okay? Now, and that's what you see here. Okay? This is the Western blotting results. Now, there is also a process called fluorescent activated cell sorters, which is right here. Because, hey, wait a minute. If I can do this with proteins, or I can do this in a little microwell, can I attach this to cells and use this to kind of separate out cells? Sure, you could. Basically, you're going to use this for diagnosing or monitoring the progression of diseases involving white blood cells. For example, HIV progression, stages of lymphomas or leukemias. What you do is you fluorescently label the antibodies. They would attach to target cells. The targeted cells have the specific target antigen. And then what you do is you run the suspension of cells through a cell sorter, which is kind of like a flow cytometer. It basically has the cells going through one at a time and separates and counts the selected number of cells along with size and density. If the antibodies are there, it says, okay, it goes left. If the antibodies are not, it goes to the center. If the other antibodies are, and it flips it over. Okay? Now, Immunoassays that involve visible antigen-antibody aggregates. Okay. Remember that antibodies can cross-link multiple cells due to the multiple binding sites. This creates a clumping effect, a meshwork of antibodies and antigens. If you remember, we had some of these tests before. Okay? And the clumping effect, sorry about this one ahead, is something that you would see with the ABO blood types, such as you see there. Now, keep in mind, you can use IgMs. You can use IgGs just as well. The big thing is that you're going to have the antibody, which has at least 2 different binding sites. It will attach to one cell and then usually attach to another, and you have these cross-referencing. So if you take a look at what you see there, what do you have? Type A blood or type B blood? Type B is not agglutinated. It's not all clumped up. Anti-A is all clumped up. So this person had type A blood. Now, there are a couple of other ones. We had direct agglutination, you have passive agglutination. This is where you use antibodies or antigen to attach to latex beads to create a larger and thereby easier to see aggregates. This is used for detection of bacteria, viruses, parasites, hormones, fungi, drugs. Precipitate reactions. The use of precipitate, basically what you're doing is you're using the detection of soluble antigens by cross-linking them to create a matrix in which the precipitate comes out a solution. Okay? The complex, though, may take some hours to form and develop only if special concentrations of the antibody and antigen are present. The best way to get the proper concentration of antigen and antibody is to allow both of them to diffuse through a gel matrix. Okay? This process is called immunodiffusion. This process allows for the precipitate to form in distinct region where the antigen and the antibody come into optimum concentration. Okay? They're basically diffusing at their own rate and speed because some particles are bigger, others are not. The region of precipitate formation is called the zone of optimal proportions, as you see in this diagram here. But you know what, you'd see it really nicely if you go to 474 and you see it in the Octoloni test. Now, it's rarely used today due to the expanded use of labeling antibodies. Octoloni techniques, the test is used to detect autoantibodies to certain connective tissue disorders, and you have a precipitate test that's based on the gradual diffusion of antibodies and antigens through a gel-filled petri dish. You can go into labs, they will practice this, but like I said, they usually have much more accurate controls. So you have an anti-X antibodies, and you have a positive control, negative control, and you have your different samples. Now, the antibodies are going to diffuse out from the center well. The other types of antigens will diffuse from those wells too. When they meet together, they're going to form antigen-antibody complexes, and you're going to have this agglutination. Now, you may have more than one on the gel. And by the way, before we go, I wanted to just make sure that you were clear about one other area that was important. And this has been something, it's gotten to the point where it's very, very common. And that is, as you can see here, this is the pregnancy tests. Now what you've got there is really a simple antigen-antibody test. What's happening is the individual takes the urine sample, and through the urine comes the HCG molecules. And as you can see, the top one is pregnant. The bottom one only has a single line. The top line, the first line there, is to identify, yes, it's passed through. And what would happen is the next line, the purplish line, I believe, is the line indicating pregnancy. So that's where you're going to have this reaction there, the antibodies, with the antigen being human chorionic gonadotropin. Okay? Well, I appreciate your patience on all of this. I want to make sure I get these all covered up. We are done with this section. This is important because it's going to give you some perspective on a variety of applications of the immune system, and gives you some perspective on issues of vaccines and other immune tests, immunity-based tests. And then I would have you plan on getting ready for Lecture 8, which is going to involve host pathogen interactions and, of course, epidemiology. And then we start moving toward closer and closer to all the different pathogens that are disease agents. This section will also give you some background for you to effectively handle the ELISA lab. Don't worry, it's not that complex, but it should reinforce some of the things you have. And I do encourage you to take the time to review the YouTube videos. And with that, I hope you have a very nice day.